Hello, my name is Oliver Bonham-Carter of Allegheny College, and I'm pleased to present our paper over our method and tool that my colleague Dr. Yimon Thu and I completed for the analysis of gene expression data using Housekeeping Gene. Our tool is called GenExSt. Our tool GenExSt has applications in cancer research. In this figure, you can see two karyotypes taken from cancerous cells. On the left-hand side, we can see a very unstable situation where we have copies of chromatids that shouldn't be there. On the right-hand side, we still see a lack of stability, but maybe less so. In both cases, these cells were cancerous. GIS genes are genes that produce a stable genome. And when you have a stable genome, then your cell development is generally natural. However, when these GIS genes are not working correctly, then the genome becomes unstable, and therefore you have the likeliness that cancers could result. So therefore, it is important that your genome has relatively low levels of instability for natural growth. Otherwise, if you have high levels of instability, then you may have dysfunction and certain types of breakdowns, which, if they're not caught by apoptosis, could develop into other kinds of disorders. It's this area of instability that the cancers could become more prominent. In healthy cells, for example, we know that there are pairs of GIS genes that work together in tandem. In this case, BRCA1 and BRCA2 has been found to work together with PARP1 to produce repairs in the cell. Now, for instance, if there's something wrong with BRCA1 2, then PARP1 appears to have to work just a little bit harder in order to produce the same repairs. However, if PARP1 is also inhibited, then there can be no repairs. And so we're saying in figure part B here that there are some healthy cells for which GIS genes work together in tandem. But what kind of work together is that? Is that a negative correlation where one gene is up and the other gene expression is down? Or are they working together where both gene expressions are up in order for certain types of relatively stable genomes to be produced? So in other words, our hypothesis is the following. Is it possible to find two different genes that appear to have a positive correlation in cancer data? In other words, is it possible to find two different GIS genes which are both working at elevated levels to produce a stable genome? Now, I use the word plausible when I say correlations, because plausible means that we are finding evidence of correlations, but that doesn't necessarily mean that they're biologically relevant. They still need to be checked in the lab. So to find these correlations, our tool comes in at this point. We basically take gene expression data, we use it to create linear models, and from those linear models we collect R-squared values, and the R-squared values are then used to create these heat maps. And the heat maps are then going to show us which are the elevated R values, or R-squared values. In that case, those are the values which appear to have a good correlation between types of genes. Heat maps were used to help us visualize these results. Now, the tool has two different versions. The tool has a Linux version, which works at the bash, and also a version that works as a browser. They both produce exactly the same results, but the browser version is designed to be a little bit more user-friendly. But again, they're exactly the same. Incidentally, you can find the repository for this project at GitHub at this link here, where you'll find both the bash and the browser versions. And we designed our tool to run in Docker containers, or a Docker container, so that when you build the container, all the important Python libraries are installed at that time. This doesn't mean that this is the only way to run it, though. You can run this outside of the container, but you'll have to install the libraries yourself. This is a list of our housekeeping genes that we use for normalization. This is a partial list of the GIS genes that we use in our analysis. Our system uses normalization to function. To find our normalization factor, we took all of our housekeeping genes and we averaged the expression values. Next, we took this value and used it to divide each of the GIS gene expression values. This value was then log transformed for enhancement. Next, we created our linear models, where each of the genes, the modified gene expressions, was then put into a test of an all-against-all variety. Every gene was then put into a linear model with every other gene. The R-squared values of these tests were then collected and poured into our heat maps. The R-squared values were used to inform us of the correlations that exist between the genes. The thinking about the R-squares is the following, that as R-squared values get closer and closer to 1, then there is presumed to be more and more correlation involved. Values which are far from 1 are likely to have very little correlation. But we must say here that when you're saying that two genes have a high correlation by their R-squared value, we're suggesting actually that those values have to be checked in a biological lab to ensure that they are biologically relevant. These are some examples of our outputs from our tool. When we use just one housekeeping gene for our normalizing factor, we found that the results were really off and they're really not plausible from what we might expect to find in biology. Here you find that many of the values appear to be 1 or close to 1. This suggests that a lot of high correlations. Remember, these values are the R-squared values, and upper values suggest high correlations. However, when we began using more and more housekeeping genes taken together as average values and then dividing those values into the GIS genes to create our R-squared values from our linear models, we found more realistic looking results. Here we find that a majority of the values is far from 1. That means that many of the R-squared values is far from 1, which is exactly what you'd expect to find in biology. The values which are close to 1, or the colors in these heat maps which are close to yellow, suggest correlations. Each of those correlations still has to be verified in a biological setting, but these are more realistic to what we would expect to find in nature. This is another trial with different heat maps or different housekeeping genes to create our normalizing factor. And here's another one. And as we can see, that when you use up to 10 different housekeeping genes to create the normalizing factors, the results become more realistic. Therefore, the take-home message from these sample tests was that when you use single expressions to normalize, it doesn't work very, very well. You get many false positives. However, when you use multiple expressions for normalization, your results look more like what you'd expect to find in nature. To have a look at the Genex software itself. Incidentally, you can go to GitHub and do a search for Genex on their website, and you can find the repository that you can clone and install on your own machine. Getting back to the software, you'll see this as your first screen when you load this in Streamlit. The first thing you're going to see in your menu options are Demo Heatmap and Demo R-Squared. Now, it's assumed that you'll be running this in a Docker container, in which all of the necessary Python libraries have already been installed for you. However, if you want to install this outside of a Docker container and install and manage the libraries yourself, then you can use this Demo Heatmap and Demo R-Squared to ensure that the relevant and correct installations have been performed so that you can run GenExSt. Click on the gene expression analysis. The first thing you're going to want to do is to adjust your threshold value. The threshold is the upper value of the R-squareds that you'll be collecting for your heat maps. I always had mine to 1.0, but you can adjust this if you need to filter your data for some reason. The next parameter you'll have to edit or enter is your instability suppressing or GIS genes. This is this file that we'll be putting right here. It's just a list of files. Next, you have your housekeeping genes to install. I call this file my norm names file, but you can call it whatever you want. Inside this file, you can even dedicate or specify different types of groups that you'd use for different normalization experiments. This is all mentioned inside the paper. After that, you click on Compute, and the magic begins. Right down here, you'll see that these are just outputs for all of your results. You'll notice that the first set of results that come out is called raw. These are just the data themselves without being transformed in any particular way. 1 and 2, these are just this is actually the data straight out of the files, out of the data files themselves. But 3, 4, 5, 6, and 7, those are your groups that you'll be using, that you'd specify in your norm file. Now, to find your files, go locally to your 0_out analysis, where you'll find your HTML. Each of these HTML files is actually a heat map, and by clicking on one, you'll get something that looks like this. We use Plotly to actually be able to interact with our heat maps in this way. Now, what we see are at the intersection of each gene pair is a color. The color correlates to, on the side of the screen here, this bar of values. And that bar of values is actually the R-squared value. So first of all, we see this line right here that goes diagonally through, which is yellow. This indicates an R-squared value of 1. But if you notice, if you look closely, you'll see that on the X and Y axis, it's actually the same gene which is being paired with itself. This is a linear test of one gene on itself, and so you see a very high correlation because it's the same gene. What you're looking for in this case is not necessarily yellows, because that's really hard to find, but instead you're looking for colors in your heat map which are close to yellow. The darker the color, the further down the line it is from the R-squared values. In other words, the worse the correlation is. The brighter the color, the better the correlation. And so typically what you're doing is you're looking for gene pairs where you find a color which is close to yellow, and that indicates a high correlation. In this case, these two genes share a correlation from R-squared of 0.825, which is not bad. If you have any questions, please write to myself. My name is Oliver Bonham-Carter, or my colleague, Yimon Thu, and we'd be very happy to answer any questions that you may have. Thank you.